This video is from the Chemistry of Life section, Biochemistry and Enzymes, under the Metabolic Pathways. We just finished talking about enzymes. Reactants for enzymes are called substrates, and when biological reactions occur in the body, there is a pathway that many of these reactions undergo. We start with a reactant, and that reactant is modified. Biological modifications are often done by enzymes. They reduce the activation energy, and they make reactions go faster by putting substrates in close proximity. Enzymes have a specific shape, like a lock and key, to certain substrates. So the substrate for enzyme 1 would be this reaction. What is produced is a product from that reaction, but it may not be the final product in the reaction we want to occur. Instead, we call it an intermediate. These intermediates have been modified in some way, but not all the way to our end product. Any number of enzymes could work in a pathway. For enzyme number 2, the substrate is this intermediate in blue, intermediate 1. The product after enzyme 2 is done with this substrate is another intermediate, intermediate 2. Each different chemical reaction has a different number of steps and enzymes working on it. Eventually, a product is formed. This is the product that we wanted and got from a metabolic pathway, a number of enzymes working on different intermediates. In order to get to that product, a final enzyme had to be used in order to change the substrate into the product. Once the body, different cellular reactions have occurred, and enough product is formed, there needs to be some kind of feedback mechanism. This feedback mechanism helps prevent more product being produced than necessary and more intermediates produced than necessary. So what happens is a negative feedback signal. Because enzymes are so shape-specific and substrates can't fit into enzymes unless the shape is correct, by changing the shape of enzyme 1, we can turn off the entire subsequent reactions. This can be done through an allosteric site. So this negative feedback turns off enzyme 1 and nothing else can be produced if there's enough product produced. Enzyme change in shape changes how it works. This can mean that we can regulate an enzyme, which is what allostery is. It also means that we can stop an enzyme, or we can change its productivity, or how much product is produced. So if we change the shape of an active site, that can happen by changing the pH away from optimum, changing the temperature away from optimum, and this could cause enzymes to denature or to change shape so significantly they don't function anymore. Other things that change the active site's shape are coenzymes. There may be a site on the enzyme in its globular 3D structure that a different molecule can fit in, and that can change the shape of the active site by not landing in the active site itself. And inhibitor molecules. Inhibitor molecules can be competitive, where they fit in the active site and block substrates from connecting, or they can be non-competitive and they can fit in a different site on the enzyme, changing its shape. That's metabolic pathways in the Chemistry of Life section on the Biochemistry and Enzymes sheet.